Hello, my name is Eric Halioua. I'm the president and CEO of PDC Line Pharma, and I'm going to present you over the next 15 minutes the new class of cancer vaccine that we have developed based on an off-the-shelf antigen-presenting cell line, which is the origin of the name PDC Line. So in terms of introductions, the company is a spin-off from the French blood bank. In fact, the scientific founder, Joël Plumat, has identified in the blood of a leukemia patient, a very rare leukemia affecting the plasmacytoid dendritic cell, which are the tiny part of the dendritic in the blood. And he was the first one to characterize this cell line. And from this leukemia and from the blood of these patients, he has developed a GMP cell line that we are still using today as a vaccinal platform. Since then, the company has been growing, and we are now a team of 24 people, including seasoned executives from biotech and pharma. We raised close to 32 million euros in equity and non-dilutive money. The three key advantages of our technology platform, and I will describe it in more details later, it's a cell line of dendritic cells, so it's extremely easy to expand. The origin of the cell, the plasmacytoid dendritic cells, induce a very high potency in terms of antigen presenting capabilities. It's a very versatile platform. We can load the cell line with any kind of antigen specific of any kind of cancer. So it's broadly applicable to any cancer. And last but not least, we're on clinical stage. A phase 1b academic clinical trial has been completed in melanoma, and we are ongoing in a phase 1/2 for non-small cell lung cancer. In terms of pipeline, as I said, we have first academic trial that has been done in PDCML in melanoma that is completed. The result has been published. We are now focusing really on PDC lung with the 6 shared antigens, tumor antigens that I will present. And we have in preclinical development a new kind of antigens, which are the neoantigens. And we would like over the next few years to enter in clinical trials with this modality as well. The team is one of the key assets of the company. We have established a very robust and experienced team. Myself, I've been co-founder of several biotech companies. I raised more than 130 million euros in my career. I've been involved in several IPO and M&A. Laurent Lévy is the co-founder with Joël Plumat. We have as well Shanna De Bruyne, that is our medical director, that spent a long time with GSK Vaccine, where she led the MAGE-A3 cancer immunotherapeutic registration trials, and Claude Edry, our VP of Pharmaceutical Operation and Quality, with a long experience in vaccine as well. The last year has been a year with strong achievement for the company. We get GMP approval for the manufacturing and quality control and release of the PDC lung in January 2019. In March 2019, we signed an important deal, licensing deal with LG Chem, the Korean company, for the development and commercialization of our lead compound, PDC lung and cancer vaccine for lung cancer. The total deal value was $123 million plus significant tied royalties on net sale in Asia. We get authorizations to launch in phase 1/2 trials with PDC lung in non-small cell lung cancer in France and Belgium. In August 2019, Dr. Shanna De Bruyne, our medical director, arrived in the company. December 19, we closed a 20 million B round of financing led by the multi-billion Asian VC KIP, Korean investment partner, very known in this area. We've administered the first vaccine with PDC lung to a patient in February 2020. And in June 2020 as well, we had a strengthening of the board of directors with the arrival of Mondaire Majoubi, an industry leader in the field of immuno-oncology. Let's go now in terms of technology presentations. So one of the key strengths as well of the technology, it's the scalability of the manufacturing process. Based on the master cell bank that has been approved, GMP approved by the French authorities, we have different bags of cells that we are using to seed bioreactors, single-use bioreactors. And after a few days of culture, the cells are in suspensions in a synthetic medium, we can produce billions and billions of cells that we load individually with peptide representative of antigens, what you see in different colors here. And then the cells are irradiated to stop the proliferations. I remind you that it is a leukemia cell line that we are using. Then we put the cells in an off-the-shelf systems, the cryovalve from aseptic technology. We store liquid nitrogens. And when the patient is ready, we just have to thaw the cells and infuse to the patients. And the cells make the job, which is priming the T cells, antigen-specific T cells in the lymph node, and expand T cells and memory T cells that go to the tumor microenvironment to make the job, which is to kill the cancer. We did different benchmark study to compare the potency of our PDC line in vitro with the conventional dendritic cells and the myeloid dendritic cells, autologous and allogeneic. And you see here by dextramer FACS, if after 21 days of culture, you see that we are 20 to 200-fold more potent than the benchmark, and showing the potency of the cell line. We did study with melané in this experience, but we did study as well with neoantigens, which is a very hot topic today. It is the antigen specific of the tumor of the patients. And we have selected two different peptides we've identified in the literature where the mutations has been clearly established, and have demonstrated is what you see on the right of the cells with the blood of 3 healthy donors that can definitely expand neoantigen specific T cells thanks to the cell line loaded with the peptide, and we can increase, the fold increase is above 150 to 200-fold for the different patients. Knowing that in healthy donors, the frequency of these T cells, neoantigen specific T cells, is extremely low. We are talking about 1 out of 1 million in the blood. The cell line is very versatile, as I said. Today we present the peptide and the antigens through the HLA-A2, which represents 50% of Caucasian populations. But we have as well genetically engineering the cells to produce additional HLA, like the A24, to enlarge the target populations, and A24 represents 50% in Asia. And in vitro, in small scale, the results are extremely interesting. We load the cells today with peptide, but we can use as well retrovirus modifications and transductions of antigen. It works extremely well. And we did as well experiments with tumor mRNA transfections. It works well as well. So we are very versatile in terms of tumor antigen loading modalities. In terms of IP, we have a very unique cell line. The master cell bank is definitely unique, coming from the blood of one donor, unique donor. It has been GMP approved by the French authorities. We have 2 patents that has been granted protecting the dendritic cell line and the applications, and a new one that is being filed in 2018 related to the genetic optimization of the PDC line. We have, of course, all the know and data to complete this IP. In terms of clinical trial in melanoma, result was interesting. It helped us, in fact, to design the lung clinical trial that is ongoing now. In fact, it has been, we infused 9 patients with advanced melanoma tumors with a cell line loaded with 4 different antigens that you see here: tyrosinase, GP100, melané, and MAGE-A3. The results were promising. It's where we have seen an acceptable tolerability and safety profile. That was the primary objective. We have demonstrated after 3 infusions that no allogenic rejections were detected, and significant antigen-specific CD8 T cell present in few patients, directly detectable in the blood of patients without any restimulations. And that despite we infused just 3 times and without any immune checkpoint co-administration at this time. And we have demonstrated later that the synergetic effect between the checkpoint inhibitor and our cell line is what you see here with the blood of melanoma patients, which is a very hot tumor with very high tumor-induced immunosuppression. You see after 1 week of culture with PDC-VAC or technology that we have 15-fold increase in terms of antigen-specific T cells. With the checkpoint inhibitor alone, we have almost, it's not representative, and twofold increase. But if we combine both the checkpoint and the PDC-VAC in the blood, the PBMC of the melanoma patient, you see this strong synergetic effect with the twofold increase. So the lung cancer, the trial we are ongoing today. So you see here on the top in black, the antigens we have selected, MAGE4, MAGE3, MUC1, NYS01, survivin, and multi-1. For each of these antigens, we have demonstrated in vitro the capacity of the cell line loaded with this peptide to expand antigen-specific CD8 T cells. Some of them are stronger than the others, as you can see here for MAGE3, survivin, and NYS01. But we have demonstrated the capacity for all of the antigens to expand thanks to the PDC line. The design of the current clinical trial is 62 patients. We will test low dose and high dose of vaccine, and we infuse 6 times. And we start with the vaccine alone in immunotherapy, then in low dose, then in high dose. Then we will go to the combination with combo with checkpoint inhibitor, low dose 6 patients, and high dose of vaccine with combo checkpoint inhibitor 40 patients. And it's in this last cohort we would like to observe clinical response of the PDC lung. In terms of funding strategy, we get this 20 million euros of funding that will provide the money required to finance the current clinical trials with the 62 patients, and to carry on the work on maximizing the value of the technology platform. And we expect in the range of end of 2022, beginning of 2023, a new finding or a deal with a pharmaceutical company for next clinical trials that could be a randomized proof of concept study, and to start new setting indications, analysis and new indications, and set up in the US and in Asia. On that, I would like to thank you for your attentions, and with the last figure, which is the team of PDC Line Pharma. Thank you.